So let's take a look at meiosis. Meiosis is basically the process by which our body is going to produce reproductive cells. These reproductive cells are also called sex cells. Another name for these reproductive cells is gamete. Now, what is meiosis? Meiosis is also called a reduction division. The reason why we call it a reduction division, it's because from a parent cell that is diploid, we are going to produce daughter cells, 4 daughter cells that are haploid. So we are going from 2n number of chromosomes to n number of chromosomes. That's why we call it a reduction division, because of the decreasing in the amount of chromosomes in the gametes. Meiosis is also 2 consecutive divisions. So it's not barely 2 divisions, it's 2 consecutive. Now, these 2 consecutive divisions do not have an interphase between the first division and the second division. Also, what I would like you to know is that meiosis is the process by which there is an introduction of genetic variation in the gametes and ultimately in the population, right? What is the process by which these genetic variations occur? We are going to address that. So, again, meiosis, we are going from a parent cell that is diploid, so it contains 2n number of chromosomes, to 4 daughter cells that are haploid, so they contain n number of chromosomes. These cells are called gametes. They are also called reproductive cells or sex cells. In the female, the cells are called eggs, while in males, the cells are called sperms. So meiosis occurs in sex cells. We have 2 divisions, so the graphic representation on your right-hand side shows you 2 divisions with a parent cell with 2n chromosomes, that these chromosomes are going to undergo interphase just like any cell, right? During interphase, you have the S phase. So the chromosome you receive from mom and the chromosome you receive from dad are going to be replicated, right? And after replication, each chromosome is going to be made up of 2 sister chromatids, right? Every single of your chromosome is going to be made up of 2 sister chromatids. So that's during interphase. And eventually, from interphase, now we are going to enter the division phase. And here we are going to have meiosis 1, which is the first division. And that meiosis 1 is going to be made up of a number of subphases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Just like during mitosis, the same terminology is used. However, we have to say specifically that the prophase is the prophase of the first division of meiosis. Then eventually, after that division, we are going to have 2 daughter cells. Now, these 2 daughter cells are haploid. And the reason why we say they are haploid is that the 2 homologous chromosomes, one coming from mom and one coming from dad, are going to end up into 2 separate cells. That's the reason why we say that after meiosis 1, we have daughter cells that are haploid. Eventually, during meiosis 2, now, we are not going to focus on homologous chromosomes anymore. We are going to focus on the sister chromatids. At that point in meiosis 2, the sister chromatids are the one that are now going to separate and end up in different cells, as you can see here. So, Again, in meiosis 1, as we start meiosis 1, the cell is diploid. After meiosis 1, the homologous chromosomes are now in two separate cells, right? Now we have haploid cells. And from there, this cell, for instance, as it undergoes meiosis 2, so prophase 2, metaphase 2, anaphase 2, and telophase 2, the sister chromatids are going to separate, and now we are producing 4 gametes that only contain half the number of chromosomes the parent cell contained. All right. So meiosis 1, we have prophase 1, metaphase 1, anaphase 1, and telophase 1. Now, the question you need to ask is the following. Well, what happens during prophase 1? What is characteristic of prophase 1? Well, during prophase 1, homologous chromosomes are going to pair up together. So if we consider, for instance, chromosome number 1 you receive from mom and chromosome number 1 you receive from dad, these two chromosomes are going to pair up. They pair up and form what we call a synapsis, right? Now, there are a bunch of proteins that bring them together and form what we call the synaptonemal complex. But I'm not going to bug you with that. Don't worry about that. So, as the two chromosomes, homologous chromosomes, come together and pair up together, now this gives the chance for the homologous chromosomes to cross legs in a process that we call crossing over. That crossing over is going to ultimately lead to exchange of the genetic information, which actually brings the possibility now of genetic variation in the gametes. Then we move to metaphase 2. In metaphase 2, the homologous chromosomes are going to align on the equatorial line. Now, as one pair of homologous chromosome and another pair of homologous chromosome and the third pair of homologous chromosomes align on the equatorial line, is completely unpredictable. So we are going to see that there are many combinations possible as to how homologous chromosomes can align on the equatorial line. This is another level of genetic variation that we are going to see in the gametes. And eventually, anaphase 1 is going to lead to the separation of the homologous chromosomes moving toward opposite direction. And eventually, in telophase 1, we're going to have two new daughter cells that each is going to have one set of chromosome from the homologous pair of chromosomes, so only one set, right? But each of the set has sister chromatids. Remember that. That's why we say each with 23 duplicated chromosomes, right? So now, going back here, and this is a graphic representation, we have a parent cell that undergoes replication. As it undergoes replication, then meiosis 1 is going to be triggered. I already told you all the different things that are happening in meiosis 1. Please review them. The cell is diploid. Crossing over is going to happen. The homologous chromosomes are going to pair up, and you can see, as you see pair number 1 and pair number 2 right here, they are paired up, right? And they're crossing their legs. Then we come to metaphase 1. And as we look at metaphase 1, if we draw a line here, we see that the way pair number 1 is aligned is different from the way pair number 2 is aligned. Now, for the sake of simplicity, only 2 pairs of chromosomes were shown here. Now, imagine in our system, we do have 23 pairs of chromosomes, right? So eventually, anaphase 1, we are going to have the pair of chromosomes now that move toward opposite direction, right? And eventually, we are going to have a depression that is basically the what is it called? I'm drawing a blank. A cleavage furrow. I have it. A cleavage furrow. As the cleavage furrow develops, eventually the two daughter cells are going to pinch off away from each other and form two daughter cells that are haploid here. Now, what is a crossing over? So you have homologous chromosome right here, right? With one sister chromatid and two sister chromatid for this chromosome that you receive, say, from mom, and this one you receive from dad. Now, you're going to have that a leg that you receive from mom, from dad right here, is going to connect with a leg from mom right here. That leaves the possibility of exchanging some genetic information here. So we have this crossing point. This crossing point, it's called chiasma. Okay? It's called chiasma. This is where the crossing over is happening. And now, let's see the result. The result is that now we have one chromosome here that is a recombined chromosome, and another one here that is also recombined. So we see that this one is a parental chromosome from mom, this one a parental chromosome from dad, and this one is a mixed, and this one is a mixed. So 2 and 3 are mixed chromosomes, right? This is basically bringing in genetic variation. Now, beyond the genetic variation, crossing over is bringing, the metaphase is also bringing a level of genetic variation through a process that we call independent alignment. So the way these chromosomes are going to align on the equator line, it's not the same way these chromosomes are going to align on the equator line, which is different from how these chromosomes are going to align on the equator line, and different from this one. So now imagine again that we have 23 pairs of chromosomes. The possibilities of alignment during interphase are just humongous. In addition to that, you have crossing overs, so that there is never two gametes that are the same. It's not possible. There is never two gametes that are the same, right? Whether you are talking sperm cells or you are talking egg cells, they are never the same. So the independent alignment of pairs of chromosomes during metaphase 1 and the crossing overs occurring during prophase 1 are responsible for the diversity that we see in the population. Now, how about meiosis 2? In meiosis 2, now we are focusing on the sister chromatids for a given chromosome, right? So prophase 2, obviously there is no interphase between telophase 1 and prophase 2. There is no interphase, so there is no replication of DNA. At that point, we are going directly a distribution of chromosomes from the previous division, right? So the chromosomes are going to condense again. In metaphase 2, now the chromosomes are going to align on the equator line, and again, now the spindle are going to target the centromeres of the chromosomes, and now In anaphase 2, the sister chromatids are going to separate and move toward opposite direction. And telophase 2, we are going to have a cleavage furrow that develops. We're going to have cytokinesis that is going to develop, and we are going to have each daughter cells at this point 4, right, are going to have 23 chromosomes each, right? And these daughter cells do not look like any other daughter cell when it comes to the genetic information. I'm not talking in terms of quantity of genetic information, but I'm talking in terms of quality of the genetic information, because technically all the daughter cells, the 4 daughter cells, they should have the exact same amount of genetic information, which is 23 chromosomes in an egg cell and 23 chromosomes in a sperm cell. All right. So a graphic representation of meiosis 2, we have prophase 2, where the chromosomes are going to condense, right? Metaphase 2, the chromosomes are going to line up on the equator line, with each having spindles attached to the centromeres, right? And the spindles are going to pull away from each other. In anaphase 2, the sister chromatids are now parting away from each other, all right? And anaphase 2, again, they part away, they keep parting away. Telophase 2, we have cleavage furrows that develop, and cytokinesis is also happening at the same time. And eventually, we have 4 haploid cells, all genetically different one from another. So that's what happened. Now, let's take a specific look at spermatogenesis and oogenesis. Oogenesis is the formation of egg cells, while spermatogenesis is the formation of sperm cells. Now, spermatogenesis happens in males. In males, after puberty, this process never stops, right? It's a continual process after puberty. About 400 million sperm cells are made per day. While in oogenesis, this is the formation of egg cells in females. The process is going to be different from what happens in males, because the process of meiosis in females starts before birth, right? It starts before birth. However, as it starts before birth, you have a number of cells that are going to stop at a particular stage of meiosis, and they stop at that stage until the age of puberty. As a lady reaches the age of puberty and the hormone production changes, now the lady is now going to be in a stage of her life where meiosis is going to continue by a female releasing 1 egg each month, right? I'm not going to take you into the details. But what is characteristic and striking with meiosis in female as well is the fact that while sperm cells are all equal in terms of size when they are produced, so from one parent spermatogonium, we are going to have 4 sperm cells that have equal ability to be part of reproduction. However, in females, from one oogonium, we are going to produce only 1 egg cell and 3 polar bodies. These three polar bodies are only there to store the genetic information that needs to be discarded. Only one egg cell can be fertilized or can move on to be fertilized, right? The polar body are discarded. Now, typically, one egg is released per month, right? About 500 eggs are going to be produced through the entire life of a lady during the reproductive cycle. Now, as I was mentioning earlier, when we start with one progenitor sperm cell, we end up with 4 sperm cells with all the ability to fertilize an egg, right? We are moving from a primary spermatocyte that is diploid to sperm cells that are haploid, and again, all with the ability to fertilize an egg. Now, how about oogenesis or egg formation? Now, the primary oocyte that are produced before birth actually are stopped at a particular stage of meiosis. And then, as the lady reaches puberty, you are going to have here one polar body, here the first polar body, and the cell that is going to become the egg. So this cell is stopped at this stage for a long time. And then when the cell is released during the cycle in a month, this secondary oocyte is going to resume the process of meiosis. And what is going to trigger the completion of meiosis? It's actually the sperm cell coming here and trying to to fertilize the secondary oocyte. By trying to fertilize the secondary oocyte, there are a number of events that are going to develop here that are going to trigger these cells to undergo the second division of meiosis. So the one polar body here is going to actually divide into 2, right? And this cell also is going to divide into 2 so that we are going to have 1, 2, and a third polar body. While this big cell, that is actually the egg, is the one that is going to move on to be fertilized. And notice that this egg only contains N chromosomes, right? And only that cell can be fertilized. And once the egg is fertilized, now we don't have an egg anymore. We have a zygote, right? All right. So let's compare meiosis and mitosis. Meiosis is the process by which gametes are formed, while mitosis is the process by which growth occurs in our body. We can repair damaged organs, tissues, cells, right? Meiosis occurs in sex cells, while mitosis occurs in body cells, also called somatic cells. Somatic cells. Meiosis, it's 2 consecutive divisions, while mitosis, it's 1 division. From meiosis, we produce 4 haploid cells that are all genetically different cells, while from mitosis, we produce 2 diploid cells that are all genetically identical. So here we have a graphic representation of what is happening during meiosis versus mitosis. So in prophase 1 of meiosis, we have synapsis of homologous chromosomes and crossing over, while in mitosis there is no synapsis, right? In metaphase 1, the homologous chromosomes are going to line up on the equator line, while in metaphase of mitosis, the chromosomes are all going to line up regardless of where they come from. Do they come from mom? Do they come from dad? It doesn't matter, right? During anaphase 1, the homologous chromosomes are going to part ways, while during anaphase of mitosis, the sister chromatids are the one that are going to part ways. In meiosis, telophase 1, we're going to have haploid cells, and these haploid cells are going to undergo another division that produce 4 haploid cells that are all genetically different, while in telophase of mitosis, we have 2 daughter cells that are genetically identical and diploid. All right, so while during cell division, we expect the genetic information to be the same when it comes to distribution, that process is not always perfect in our system when cell divisions occur. There is a dysfunctioning in cell division that can occur at times. One of them is called non-disjunction. Non-disjunction can occur during meiosis, whether it happens during meiosis 1 or meiosis 2. Now, the consequences are more severe when non-disjunction occurs during meiosis 1, because during meiosis 1, if non-disjunction, which means that the homologous chromosomes do not separate, when that happens during meiosis 1, all daughter cells are not going to contain equal amount of chromosomes, right? We're going to have 2 daughter cells that have too many chromosomes and 2 daughter cells that are missing some chromosomes. Now, non-disjunction can also occur during meiosis 2. And when that happens, 2 daughter cells are going to be perfectly fine, while 2 daughter cells are not going to be fine. One is going to have too many chromosomes, and the other one is going to have too little chromosomes. Now, when non-disjunction occurs, this leads to a condition that we call monosomy and another condition that we call trisomy. Monosomy means that the gamete is missing one chromosome, right? While in trisomy, it means that the gamete is having an extra chromosome, right? One example that we know, it's trisomy 21, right? Which is responsible for Down syndrome, right? We are going to address some examples of monosomy and some examples of trisomy. All right, so non-disjunction can occur during meiosis 1. When this happens, you're going to notice that all the 4 haploid cells are not containing the proper amount of chromosomes, because this one contains 2n plus 1, okay? This one contains 2n plus 1. This one contains 2n minus 1. This one contains 2n minus 1. So these 4 chromosomes are abnormal, okay? Now, non-disjunction can happen during meiosis 2, as you see here. So when that happens in meiosis 2, what we do see is that, well, 2 cells are normal while 2 cells are abnormal, okay? This cell contains 2n plus 1, while this one contains 2n minus 1. Now, when I say plus 1, this is a case of trisomy, and minus 1, this is a case of monosomy, right? Here we have two cells that are monosomy and two cells with trisomy. Okay, so all the cells are abnormal, while the cells are normal here. This one, trisomy, this one, monosomy. All right. So, now, what we notice is that in males, the cells are XY, while in females, in human, the cells are XX. Now, in the case of female, being XX, it's actually a duplication of the genetic information. For that duplication of the genetic information to not have a consequence in a female, there is one of the X's that is going to go silent at some point, right? So in a female with two copies of X, right, one of the X's is going to go silent during the embryonic development stage. That X that undergoes silence, we don't know exactly which of the X is going to be silent. Is it the X that the lady received from mom? Is it the X that they received from dad? We don't know. And what is interesting is that in the embryo, because you have many cells, not the same X is silent in all the cells of the embryo. So, which means that some embryonic cells are going to have an X that is silent that comes from mom. Some embryonic cells are going to have an X that is silent that comes from dad. And by the way, that silent X chromosome that is completely compact and very small is called a Barr body. That Barr body will not be able to express, right? And this is how female are going to compensate for having two X chromosomes. And by the way, there is a consequence to one of the X condensing into a Barr body. This is particularly true for features where the genes are actually in forms of alleles that are one being dominant and the other one recessive, right? In the case of heterozygous individual, this can have some consequences in terms of phenotypes. Now, in terms of extra chromosomes, this individual right here on the photograph is actually an individual with trisomy 21. So we can see on the karyotype here, an extra chromosome right here. And when looking at the chromosome 21, we see that the features that are responsible, that is responsible for the features that are typical to people with Down syndrome is actually a particular gene right here on the chromosome that is called the GART gene. That GART gene is responsible for the many features that we see in people with Down syndrome. Now, let's take a look at some cases where we have either too many chromosomes or too little chromosomes when it comes to sex chromosomes. One situation is called the Turner syndrome. The Turner syndrome are individuals that are XO, X, only one X, but no second X or a Y. These individuals are females. They're going to typically have a short stature, broad shoulder. They're going to not typically have menstruation, right? They're going to have underdeveloped female features. The Klinefelter syndrome, it's another syndrome that is associated with sex chromosomes. In here, the individual is going to be double XY. The double XY is definitely a male individual, that is going to have underdeveloped sex organs. Because of double X, they're going to tend to have high development of breast and large hand, long legs, and large long arms. Other syndromes, the poly X in female. In the poly X in female, we have two kind of poly X's. We have triple X and four X's. In the triple X, the female is normal. They're just going to have exaggerated female features. While in the four X's, we're going to have severe mental retardation. Another too many sex chromosomes. This is going to be in males, the Jacobs syndrome. In the case of Jacobs syndrome, we have XYY. In the XYY, the individual is going to have male features that are going to be exaggerated. They're going to be very tall. They're going to have persistent acne. Sometimes some are going to have speech issues and reading issues. Now, beyond that, the chromosomes can be modified during the cell cycle. Now, when these chromosomes are modified, there are a number of consequences that are going to happen. Some modification are going to be the deletions. Deletions is when there is loss of some genetic information. Another kind of modification of the chromosome is translocation. Translocation is basically when on a chromosomes there is a change of genetic information that swap from one position to another position. I'm going to show you the outcome in a minute. Then we have a duplication. In a duplication, the genetic information appears twice on the same chromosome, right? While in an inversion, you don't simply have a swap of information, you have a swap of information toward opposite direction. I'm going to show you actually a graphic representation what this looks like. So here we have A that is deleted, that is loss of genetic information, that has tremendous consequences. Here, a duplication, we have DE that are duplicated in the chromosome. In inversion, you have C and D here that are aligned properly, while on this chromosome here we have D and C. They are inverse. In a translocation, we have basically a change in the genes on different chromosomes, right? Which is not a good news. What is the process by which inversion is happening? It's actually this process here, right? Where the homologous chromosomes are exchanging some genetic information so that we have exchange of genetic information. So basically, this is going to conclude our lecture today. Thank you for your attention. Until I talk to you again, take care. Oh, well, actually, we do have a deletion here, translocation, and that's it.